Ion Exchange Chromatography. Ion Exchange Chromatography, or IEC, is a method used to separate ions based on their affinity to an ion exchanger. This technique is widely used in the purification of proteins, amino acids, and even water softening. Here we have a column packed with a specialized material called an ion exchange resin. The key to this resin is that it contains charged groups. These groups can either be positively charged or negatively charged. And, depending on which type is used, the resin will attract and bind ions of the opposite charge. There are two types of chromatography: cation exchange chromatography and anion exchange chromatography. In cation exchange chromatography, positively charged molecules, cations, in a sample are separated based on their charge. The process begins with the stationary phase, which is the solid material inside the column. This stationary phase contains negatively charged groups such as sulfonate or carboxylate, usually on a resin or gel. To start, the column is equilibrated with an initial buffer that typically has a low concentration of NaCl. This buffer helps control the pH and salt concentration, setting the conditions so that positively charged molecules in the sample will bind to the stationary phase. When the sample is loaded onto the column, the positively charged molecules are attracted to the negatively charged resin and bind to it, displacing the Na which was present in the initial buffer. Negatively charged or neutral molecules flow through the column and are eluted. Now you need to elute the protein of interest that is binded to the resin. For this we will add an elution buffer that will break the salt bridges between oppositely charged molecules. Elution buffers work by either increasing salt concentration or adjusting the pH to release the bound molecules. In cation exchange chromatography, raising the pH can alter the charge of positively charged molecules, making them less likely to bind to a negatively charged resin. This change in charge reduces the affinity of the molecules for the resin, leading to their elution. In the other method, the concentration of NaCl in the buffer is gradually increased. As more Na plus ions from NaCl enter the column, they compete with the sample's cations for the negatively charged binding sites on the stationary phase. This causes the bound cations in the sample to be displaced, or eluted, from the column. Molecules with weaker positive charges elute first, while those with stronger positive charges elute later, as they require a higher NaCl concentration to be released. As the NaCl concentration increases, an elution graph is generated, showing peaks that correspond to the elution of different molecules over time. Molecules with weaker charges appear in earlier peaks and are eluted first, while those with stronger charges elute later as the NaCl concentration reaches higher levels. In contrast to cation exchange chromatography, anion exchange chromatography has a stationary phase containing positively charged groups such as quaternary ammonium, usually on a resin or gel. The column is equilibrated with a low salt buffer, allowing negatively charged molecules to bind to the positive stationary phase, displacing the chloride ions which were present in the initial buffer. Positively charged or neutral molecules flow through the column and are eluted. Just like in cation exchange, we can elute the protein of interest by either changing the pH of the solution or raising the salt concentration. In anion exchange chromatography, decreasing the pH can alter the charge of negatively charged molecules, making them less likely to bind to a positively charged resin, causing their elution. Increasing the salt concentration causes chloride ions in the buffer to compete with the bound anions in the sample, gradually displacing them based on their charge strength. Molecules with weaker negative charges elute first, followed by those with stronger charges as the Cl concentration increases. Please like, share and subscribe, and don't forget to hit the bell icon for more videos.